Hello, everyone. Welcome to today's Genomics England Research Seminar. Unfortunately, Henry Holden couldn't make it today, so I'm standing in instead, and I have the honor of hosting our speakers today. My name is John Griffin, and I'm a scientific manager here at Genomics England in the research management team. So many of you will know me anyway through our interactions with the research community. We have a really exciting lineup today for Rare Disease Day. We have two rare disease talks. First, Wei Wei is going to tell us about the importance of nuclear embedded mitochondrial DNA from her work in the 100,000 genome sequences. And then William Macken is going to talk about the value of multidisciplinary research in improving diagnosis rates. So really excited about that. Before we jump into that, though, there's a couple of the usual kind of housekeeping and announcements that I would like to share with you. The first is that, of course, this seminar will be recorded. We record all of these research seminars because we can put them up on our YouTube channel, the Genomics England YouTube channel later on. So just so you know and are comfortable with that. And as I say, all of those talks are on our YouTube channel. There's a lot of really interesting stuff there. So if you haven't checked it out, you might want to take some time and have a look when you have a chance. The other thing, so before I get into the career workshop, I should also say that there are actually two kind of chat boxes. Just to remind you, there's two chat boxes in this Zoom. At the bottom, there's chat, which is great. You can just share a conversation amongst everybody at the talk. But there's also a Q&A tab. And if you go in there, that's where you leave specific questions just for the speakers today. So please try to keep the Q&A box specifically for questions for the speakers. And yeah, so what I wanted to remind you about before we start, there's a call for abstracts for our early career researcher symposium. The deadline is actually this Friday on the 3rd of March. But if you are an early career researcher, so somebody, an MSc student, a PhD student, or even just within 5 years of getting your PhD, please do consider submitting an abstract. I think it's going to be a great research seminar. And it's a really nice opportunity to just show your work and kind of network and get a bit of a exposure in the community. The other thing that we would draw your attention to is just today, the new genomic medicine service data release became available. So now there are just shy of 6,000 more genomes available in the RE. And so if you're interested, of course, you can go log in and explore that a little bit more. And then finally, our next research seminar will be on the 28th of March. We have one speaker lined up so far, who is Shadi Basuni, and he's going to talk about the importance of P53 sequencing in prognosis for head and neck cancers, which sounds great. And the second speaker is yet to be decided, but we will let you know about that as soon as possible. So I think that's it for me, actually, probably enough. And I guess we can jump right in with the talks. We're going to start with Wei Wei, who worked at Cambridge for many years, for several years with Patrick Chinnery, and did some really interesting bioinformatic work there, which I think is probably where the work, she can correct me if I'm wrong, where the work today comes from. And so she's going to tell us about nuclear embedded mitochondrial DNA sequences from the whole genome sequences in the 100,000 Genomes Project. And so for that, I'm going to hand over to you, Wei, and thank you for joining us.
Thank you, John. Then let me share my screen. Can everybody see my screen well?
Yeah, I think so. I can see it.
Okay, great. So thank you for the opportunity to discuss some of the work I did during my postdoc with Professor Patrick Chinnery in Cambridge University. So today, the project I would like to discuss is around nuclear embedded mitochondrial DNA sequence from 100,000 Genome Project. I'm going to start with a very brief background of mitochondrial and mitochondrial DNA. So let me get my printer out. So this is a cell, then here is a nuclear. So mitochondrial are special compartment in our cell. They are best known as the powerhouse. So mitochondria can break down the food molecules and turn out ATP to support provide energy to the rest of the cell. There, in a cell, there are 2 copies of nuclear DNA and hundreds of thousands of mitochondrial DNA. Each mitochondrial DNA is a double-stranded circular DNA, includes 13 coding genes, 22 tRNAs, and 2 RNAs. So majority of the mitochondrial DNA were encoded by nuclear genes. We inherit our nuclear DNA from both mother and father. The mitochondrial DNA is a maternal transmission, so we only inherit our mitochondrial DNA from mother. Then, if a mutation occurs in all the mitochondrial DNA in a body, then we named it, we call it mito-TDE homoplasmis. Then if mutation occurs in proportion of the mitochondrial DNA in a body, then we call it heteroplasmy. So this is a very brief background of mitochondrial DNA. So how we started this work to look at nonce in 100,000 genome. I think it's really kind of mainly driven by this paper published in PNAS 2018, back to nearly 4 years ago. So in this paper, they reported biparental inheritance of mitochondrial DNA in 5 families. And now I show you an example of a 3-generation family they reported in the paper. So each bar represents each mitochondrial variant. The y-axis is allele frequency. Then what they found was, if we look at grandfather, it inherited around 40% of the mitochondria from his father in blue, then 60% of mitochondria from his mother in pink. Then even more surprisingly, they said, okay, this grandfather further passed his paternal mitochondrial DNA to his daughter and grandkids. So this is a very controversial study. Quite a few comments published in next couple of months. So in our lab, we discussed it. Then at that time, so we had around 12,000 trials from Genomics England rare disease program. Then we were thinking, if this paternal or bioparental inheritance of mitochondrial DNA is real, we must see it in this large data set, or is extremely rare event. Probably we don't need to worry about it too much. Then we started to develop a pipeline that we wanted to look for the same pattern as the paper reported. Now we did see 7 families in our data set. They carried almost identical pattern as the paper reported. I show you example here. In this family 6, so this is father's mitochondrial variant mother, then in both proband and sibling, they carried around 25% of mitochondrial DNA, same as their father, then 75% of mitochondrial DNA from their mother. So we were starting to kind of look for the alternative explanation. Then our hypothesis was this nuclear mitochondrial DNA segments nonce can mimic the paternal inheritance of mitochondria. Then we developed another pipeline to detect the nonce from a whole genome sequence. I will show you how the pipeline works later. But here in this study, what we found was in these seven families, both father and the kid, they carry mega concatenate nonce. So it shows multiple mitochondrial, they concatenated together, then insert into nuclear genome. So it's it's a combination of nonce and the real mitochondrial DNA alleles created the false impression of a paternal inheritance of a mitochondrial DNA. Then after that, we published this work in Nature Communication in 2020. So from this study, we got very interested in nonce. Then we decided to further explore nonce in the whole 100,000 genome project data set. This number is really back from 3 or 4 years ago. Now, Genomic England data set had much more samples than this. But then we published this work in Nature last November. So the rest of my presentation, I will show you some results we published in this work. Then this slide is the bioinformatics pipeline, and how we detect nonce from a whole genome sequence. So here, this is mitochondrial DNA, then here is a nuclear genome, then mitochondrial DNA insert into nuclear genome, then something look like this. Then firstly, we extracted all the discordant reads, one pair of reads, one end mapped to nuclear genome, then another end mapped to mitochondrial genome. Then to look for the breakpoints between nuclear and mitochondrial DNA, we extracted the split reads. One read part mapped to nuclear genome and another part mapped to mitochondrial genome. Then we further validated our nonce use long read sequencing. So a very long read can cover the whole mitochondrial DNA insertion. This is a very high level description of our bioinformatics pipeline. Then what we found in this study, so from more than 53,000 rare disease whole genome sequence, we identified more than 1,600 different nonce. So they are showing in this circles plot. Then on the left side is mitochondrial DNA. Then on the right side, they are different nuclear chromosomes. Then in the middle, each link represents each nonce. So one piece of a mitochondrial insert into nuclear genome. Then from here, we can see the nonce involved in the whole entire mitochondrial DNA sequence and across the different nuclear chromosomes. Then we observed nearly every single person carry at least one nonce. Then average one person had around 5 different nonce. Then we further grouped the nonce based on the frequency in population. Then more than 96% of the nonce, they are extremely rare in the population. Then we also found most of the nonce we identified in this work, roughly 95%, they are novel, they have never seen before. So here showing you the lighter color in each plot, in each donut plot. Then the size of the nonce, most of the nonce, roughly 60%, they are short insertions below 200 base pair. Then the size of the nonce, they negatively correlated with frequency, nonce frequency. So this result indicates the selection against the larger nonce. Then from nearly 10,000 trials, we were able to estimate de novo nonce mutation rate. Then we found 3 different de novo nonce. They are only present in offspring, and we didn't see them in either of their parents. Then this gave us a germline nonce mutation rate is around 1 in 4,000 births. Then move to cancer nonce. What about the cancer nonce? Then generally, the tumor samples, they carried more nonce compared to their matched normal samples. Then here we were more interested in the semantic nonce. Then we used a very stringent criteria to define the semantic nonce, which is only present in cancer, not in any germline in both cancer and rare disease data set. So using this criteria, we defined 379 cancer-specific semantic nonce from 251 tumors. And it shows in this circles plot, again, on the left side is mitochondrial DNA, and the right side, different chromosomes, each short red bar represents each nonce. It's involved in the whole mitochondrial sequence across different chromosomes. Then cancer semantic mutation rate is around 3.5 multiple 10⁻², which is much higher than the germline de novo nonce. Then for the semantic nonce, they were not the same in different cancer type. Then on the top plot here shows you the proportion of the tumor samples they carry at least 1 semantic nonce. Then the bottom plot shows the mean number of tumor specific nonce. Then what we found here is some cancer type, for instance, bladder, breast, they carry more nonce compared to other cancer types. From our data set, we were trying to look for some evidence to better understand the mechanism of the nonce formation. Then here I want to show you some of the results we found in the paper. So then firstly, we asked if there is any association between nonce and different genome regions. Then here, this plot shows each column is different genomic regions. Then each row represents different type of nonce. It's from germline, or from cancer, or from different frequency of in population, rare or common. Then the color code is the significant, is the enrichment p-value. For example, for rare, common, and rare nonce, we didn't see any common and rare nonce in coding region. Then they are enriched in intron region. So this result is consistent with nonce being under evolutionary constraint. Then next, we looked at, so it has been reported nonce are involved in double-strand DNA break repair in different species. So what we looked at here is we measured the distance between NANT breakpoints to PRDM9 gene. So this gene is implicated in double-strand break repair, and this gene is also determines the recombination hotspot binding sites during meiosis. And from our data set, we found the germline NANTs, they are enriched in within 3,000 base pair of DM9 gene binding sites. Then for semantic mutation, enriched in 1,000 base pair PRDM9 binding sites. And this result added the further evidence NANTs play a role in double-strand DNA break repair. Then next, how the DNA fragment insert into mitochondrial DNA fragment insert into nuclear genome. Then from our data set, we found more than half of the NANTs, the mitochondrial DNA insert into nuclear genome through microhomology medium and drawing. Then we also saw some evidence support blur and drawing, and one small piece of insert into breakpoints region. So here we kind of found a different found that NANTs are involved in different mechanism, but overall the NANTs mechanism, they are related to genome instability. So here I want to show you one extreme example we found in cancer genome. So in this example, so here is one chromosome 5. Then each blue dot they represent each NANT. Then here is y-axis is the sequence depth around this region. So what we saw here is this circles plot mitochondrial DNA. They chopped into small pieces, then they insert into chromosome 5 multiple locations, and also coupled with the nuclear structure variation. So chromosome 5 also chopped into small pieces and lots of inversion here. Then also involved some translocation between different chromosomes. We only see in this event in cancer samples, so we didn't see it in normal sample, paired normal samples. So this kind of extreme rearrangement has been seen in chromosome. Okay, this is all I want to discuss today. So today I only have 20 minutes. We have a lot of more interesting results in the paper. If you are interested, please do check out the paper. So finally, I would like to thank everyone in my postdoc lab, particularly my PI, Professor Patrick Chinnery. Without his great support, none of this work is possible. I also want to thank all the collaborators, so particularly the Genomics England long read sequence team. They did all the long read sequencing to support our project, the validation. Then finally, I want to thank Genomics England help desk team and our local manager Peter. They have been very, very helpful through the whole project. Then finally, I thank you for you listening. Then I would like to take any questions. Thank you.
Thank you, Wei. That was really interesting. It's fascinating. Let me just take a look here. We'll look in the question box. We'll see if there's any questions coming up. There's one about the, okay, so that's probably not for you, about the newborns cohort. Or maybe it is. So, okay, so sorry, let's say the first question. I don't know if you know this. Does the newborn whole genome sequencing project include sequencing mitochondrial DNA? Probably.
I think as long as it's a standard whole genome sequence, it would include mitochondrial DNA.
Okay.
And normally for whole genome sequence, the nuclear genome is kind of a 50, or let's say 50x depth. For mitochondrial, you can get thousands depths just because the mitochondrial is multiple copy in a cell.
Okay, that's great. And thank you for the question, Shelley. Cassie asked, for the rare disease cohort, did you look at any NUMPs that potentially disrupt a gene region that could be causative recruited condition for the participant?
Yeah, that's a very good question. We did look at it, but for the rare disease, we didn't find a very convincing result. Not it cause a disease, but for the cancer genome, we found one fusion genes it potentially can cause the cancer, but for rare disease, we didn't see any. I think it's going to be a very kind of a rare event.
Okay, yeah. And then maybe we just take one more this time. Let's see. Or two. So Andrew asked, what proportion of NUMPs are thought to be functional in generating mitochondrial proteins? You know?
So proportion of NUMPs, I think if I understand this question clearly, it means if the NUMPs expressed in
Yeah,
genome.
so yeah, if they make an actual protein, if they still make a mitochondrial relevant protein.
You know, that's something we would love to look at, but from the whole genome sequence data, it's very tricky for us to look at it. We did kind of try to tackle this question from the methylation analysis. For the long read sequencing, we can detect the methylation. What we found in the paper is the NUMPs, they are higher or highly methylated compared to the real mitochondrial DNAs. So potentially they kind of they won't be expressed in the nuclear genome. But yeah, that's something we would love to look at further.
Okay. Alistair had a similar question kind of following on from that to ask if NUMPs are expressed at the RNA level. Do you know if they're?
I think if we have some RNA sequencing data, we can look at it.
Okay.
Yeah, but only from whole genome sequence data, that's something we are not able to.
Yeah. Okay. And then we have a final question, I guess, here before we move on from Jeeb, which is, there are some sequencing methods where you can just capture mitochondrial DNA only. Do you think this would help get rid of NUMPs? Do you want to get rid of NUMPs?
Yeah, I think they do. They have some kind of people develop a different technique, only capture mitochondrial DNA to target mitochondrial DNA sequence. It just get rid of NUMPs because these NUMPs will kind of bias you result if you look at mitochondrial heteroplasmids. Yes, they do. People do this special technique to look at mitochondrial DNA only.
Okay. So those, I think, are the end of our questions, and we'll move on in the interest of time. But thank you very much, Wei Wei. It's a really fascinating talk and a beautiful paper, I have to say. So
thank you very much. Thank you.
Okay, then we will move on to our second speaker, who's William Macken. William, I think, trained in medicine in Dublin and London, specialized in pediatrics, he is currently, a specialist registrar in clinical genetics with a research interest in neuromuscular diseases and in particular mitochondrial diseases, I believe. And so I'll hand it over to you, William. Thank you for joining us.
No problem. Thanks very much. Can you see my screen okay there?
Yeah, it's good.
I'm just need to minimize. Yeah, okay, cool. So can you see my pointer?
Yeah, I see it as the, yeah.
Okay, great.
Yeah.
Okay, so hi everyone. Yeah, my name is Will, and I'm talking to you today about specialist multidisciplinary input, which maximizes rare disease diagnoses from WGS data. And so this is based on a paper that we published relatively recently in Nature Communications. So I'm a research fellow just finishing a PhD in Queen Square, the Institute of Neurology in UCL, focusing on mitochondrial disease. And I'm supervised by Rob Pitcatley and Mike Hanna, who are academic neurologists who work on mito disease, and Jana Vandrukova, who's a geneticist and bioinformatician interested in neurogenetics kind of as a whole. And you'll be hearing a little later from our colleague and co-author Anna Sarkozy from GOSH about a multidisciplinary approach more broadly in neuromuscular conditions. So just to pull things back a bit, there's a long history of mitochondrial disease research in the UK and in London. So we work at the National Hospital for Neurology in Queen Square, and a lot of the lead investigators when genetics was really taking off were based in our unit. So the late Professor Anita Harding and John Morgan Hughes with Ian Holt described the first ever cause of a mutation causing mitochondrial disease. So they had these patients who had big deletions forming in their mitochondrial DNA, and this was published back in 1988 in Nature. And so today we've got a highly specialized service, so an NHS service for this group of diseases. We've got a large MDT of doctors, nurses, physios, and different types of scientists. And we offer clinics, clinical trials, genetic diagnostics. Our neurogenetics laboratory is based in the North Thames hub, which is in GOSH. We do neurometabolic investigations for the functional implications of these mutations. We have translational research, and we do a lot of MDT support. So if patients are sick in hospitals around the country, we can offer advice, and we also give advice for genetic results and interpreting genetic variants. And we've got partner sites in Oxford and Newcastle, so it's truly a national initiative. So you've heard from Wei Wei already, but briefly, primary mitochondrial diseases are genetic disorders where you get perturbed oxidative phosphorylation or other aspects of the mitochondria. And we know mitochondria do a lot of things, so there can be a lot of different symptoms. They arise from mutations in either our mitochondrial DNA or nuclear DNA. And Wei Wei touched upon the issue of heteroplasmy. So you can have a mixture of wild type mitochondrial DNA, which look like little plasmids, or ones with mutations. And because these mutations can occur only in a small number of copies of our mitochondrial DNA, you need to do really deep sequencing of mitochondrial DNA to make sure you'll pick them up. You also have to look at nuclear DNA as you would with any rare disease. We know the prevalence is about 1 in 4,300, but this is probably a slight underestimation because the carrier rate on NOMAD of pathogenic mitochondrial DNA variants is thought to be about 1 in 250. There's lots of different possible manifestations of disease. In adults, we tend to talk mostly about brain and muscle involvement, but any organ can really be involved. And in terms of diagnosing these conditions, well, to date, it's been quite complex. And I'm not going to go through every part of this process, but suffice to say, you have to look at mitochondrial DNA, usually first in blood, and also nuclear DNA with gene panels, or sometimes on a research basis with whole exome sequencing. And often you'll also have to do sampling of another tissue, especially muscle, looking at the functional implications of these mutations, but also looking for heteroplasmic variants that aren't present in blood and which are only in post-mitotic tissues like muscle, and looking for rearrangements with a dedicated approach. So all of this kind of rigmarole has led a couple of people, including Wei Wei's team, to suggest that we should be using whole genome sequencing earlier in the process, because you can look at the nuclear and the mitochondrial genomes at the same time. And there's been lots of exome studies done. There's been some WGS studies and RNA-seq studies, and the cohorts are all a bit different. But suffice to say, we're never getting 100% diagnostic rate. There's always lots of patients who are undiagnosed, and there's lots of reasons for this. But we think that part of the problem is the filtering we do. So to pick up low-level heteroplasmy, you can't use your normal variant allele fractions that you'd use for heterozygous and homozygous variants. You need to use an approach a bit more like a somatic variant caller to pick up low levels of VAF. You also might be missing diagnoses because there's a lot of phenocopies of mitochondrial diseases, Lots of diseases look like mitochondrial diseases. You might have two diseases coming together to form a compound phenocopy. And also some disease processes have secondary effects on your mitochondrial functioning. So you might actually get changes in your mitochondrial enzymes, which aren't from a mitochondrial gene. And then, as with any neurogenetic or rare disease condition, there's lots of cryptic variants that might be playing a role that we find hard to analyze. So in terms of our patients in 100,000 genomes, we had 102 patients from our clinic in this project. And what we've broadly done for the project, for the presentation I'm giving today, is that we catalogued all the patients that were included, described the diagnoses that were made with a routine approach, and then we've re-evaluated all the cases to try to address the factors that might be contributing to overlooked diagnoses. Now, I'm sure a lot of people in this audience will be quite familiar with the general process for rare disease patients in 100K. One thing I wanted to point out is that the approach uses a virtual gene panel, and so the panels looked at can vary between patients. While they'll all have had mitochondrial disorders looked at, they might have had additional panels looked at or not, and it's not an agnostic approach that's used. So after the sequencing had been done and the basic bioinformatic processing, data was filtered for rare protein altering SNVs and indels, and panels were applied, which were decided in advance, and then these mutations were reviewed by a clinical scientist. And any good candidates they brought to an MDT with the clinicians, and we made some routine diagnoses there, and that was done across all rare diseases across the country. We did some additional work where I reevaluated all the cases with no primary findings or which hadn't got a diagnosis. We did some additional bioinformatic analysis then and added in some additional panels. And then I re-reviewed all of those cases again to try to come up with good candidates to take back for formal classification and validation with our clinical scientists. So in terms of our results from the routine analysis, it was a little bit disappointing with only 16.6% of our patients getting a diagnosis. And I just wanted to point out that that wasn't really a function of the cohort, and that these were really good patients with good clinical phenotypes. We do something called Nijmegen scoring sometimes in mitochondrial disease, where you look at the different elements of the phenotype and collate them together to decide how likely it is you have a PMD, a primary mitochondrial disease. And most of our patients either had a definite or a probable PMD based on their phenotypes. So they were quite likely to have something that could be found. And there's a slight sex bias in the cohort. And one thing that's worth pointing out is that we mainly have an adult clinic. So we had a lot of singletons recruited, and they're obviously harder to resolve than, say, children who come in a trio. So these were the diagnoses that were made with the routine approach. And I've highlighted in green the ones that are true primary mitochondrial diseases. And we had quite a mixed bag apart from the PMDs, and the biggest group apart from PMD was neurogenetic and neurodevelopmental disorders, which makes sense because a lot of the phenotypes we see are brain focused. So what about this big group that weren't diagnosed? Well, from doing our review, we were able to diagnose another 15. And then in addition to those, we had another 4 patients where we had a strong VUS, which we think is the diagnosis. But at present, there hasn't been, for example, enough patients published for us to get this over the line. And then we found some patients with novel genes, and they're currently involved in functional studies, collaborating with other groups around the world. So we've pushed up the diagnostic rate from 16.6%, if we include everything, to closer to 40%. So it is a significant uplift for our patients. And I wanted to touch on the diagnostic themes. Why did we miss some of the diagnoses, and what should we do differently in the future? So we did a lot of additional work on new gene panels. So that might be based on the phenotype. So adding in another gene panel, for example, intellectual disability, if we thought it was relevant. But we also lowered the thresholds to include genes in these panels. So Genomics England uses a program called PanelApp, which is crowdsourced list of genes linked to each phenotype, and genes with a weaker association, which wouldn't meet the thresholds for inclusion in clinical testing, have an amber status. So we did include those genes also when we were looking for missed causes. So that was the biggest contributor. We also found that we missed intronic second hits in 3 patients. For example, one patient with MYH2, I found a second hit -17 into an intron, and we found that that had an effect on transcript levels. We also did a lot of work on revised modes of inheritance filtering. So with primary mitochondrial disease, we can have matrilineal inheritance, which is the most common inheritance in adults, because PMDs in adults are usually caused by mitochondrial DNA variants. But any other form of autosomal or X-linked inheritance can also happen. So we had a confusing family here where the proband shown in blue, he has deafness, and a number of other family members have deafness. He also had a movement disorder, and a number of family members had a cardiomyopathy. And by focusing on different elements of the phenotype, we were at least able to identify a dominant deafness mutation here. So it's likely that there's a number of different neurogenetic or genetic conditions happening in this family that aren't appreciated when you try to take the phenotype as a whole. We found a mosaic variant in this patient who had a mosaic variant in Dynamin 2, and she had actually been tested for this gene multiple times, but the nice coverage, I suppose, that WSGS allowed us to pick this up. We also found some patients who had missed heteroplasmic variants in mitochondrial DNA, and by using a better approach to variant allele fraction filtering, we were able to pick these up. We did a lot of reverse phenotyping in this study. So for example, we had one patient who didn't have a diagnosis. She had mitochondrial changes on her muscle biopsy, but actually seeing her in clinic, she looked a little bit dysmorphic, and there was a suspicion that she might have some minor skeletal changes. And we did a skeletal survey in her and found that she had these elongated fingers and unusual femoral necks. And so we found a variant in KIF22, which causes spondyloepimetaphyseal dysplasia. And so she probably has a mild variant of this and not a true mitochondrial disorder. We did some work on updating the literature review to help us interpret variants as well. And so, for example, in this lady who has seizures, we found that she had a mutation in COL4A2, which traditionally was only associated with something called porencephaly. We get these kind of massive cavitations in the brain, but actually now, more recently, it's been described to cause mild white matter changes and seizures. So that would fit with this patient. So I'm going to come towards the end of my talk now and just tell you in a little bit more detail about one family. So this family had 2 affected siblings, and the elder sibling is now actually, I think, 51. So they've been waiting years and years and years for a diagnosis, and they've had every type of biochemical and DNA testing done that you can imagine. And they had a childhood onset condition, and they've complex 2 and 3 deficiencies. So they had no diagnosis from the initial 100,000 genomes analysis, but I applied a metabolic panel as well and found they had a variant in mColon1, which is associated with a lysosomal storage disease. But actually, if you look at what the protein does, it encodes a calcium channel that's involved in shunting calcium between lysosomes and mitochondria. So it's very much could be causing a problem with our respiratory chain. I found a second intronic hit, which just at the time as we were doing this work was published as causing aberrant splicing. And really interestingly, apart from having a severe neurological phenotype, the younger sibling, the gentleman, this family, sadly passed away from a large gastric tumor. And we know that mucolipidosis type 4 causes a lack of hydrochloric acid in the stomach and elevated gastrin. And this was an unusual tumor in that it was very large but slow growing and indolent. And we think that this might be playing a role in creating an environment in the stomach that is pro-neoplastic. And so this might explain why this gentleman has a tumor. We've also found that his younger sister has polyps as well, so she might be at risk of something similar. So they probably, older people with this type of disease probably need to have screening for this type of neoplasia. Our work had lots of different management implications for our patients. So some qualified for new drug trials, malignancy screening, as I've mentioned. 2 patients could have their screening stepped down because they don't have a PMD. They don't need to have, for example, echoes done regularly or diabetes testing. 5 of our families had new options for reproductive screening because they never had a genetic diagnosis before. And then we needed to step up multi-system screening in some families as well. So to conclude, we've boosted the diagnostic rate significantly. And I think this suggests a new model for whole genome sequencing interpretation in healthcare. It's our feeling that the one-size-fits-all way of approaching this doesn't really work for our more complicated patients. So after the initial variant analysis, if there's no diagnosis made, we need to rethink our analysis of the data and maybe redo our bioinformatic processing, and then get someone with expertise in genomic medicine to go through these variants and decide if functional work, for example, from a research laboratory, or clinical work with reverse phenotyping or testing other family members could get us a new diagnosis. Ultimately, confirming these new diagnoses improves our patient counseling and management, as we've seen. So I think that's a really important thing to think about, not only in mitochondrial diseases, but in every rare disease. So thanks very much for listening to me. And I think Anna is just going to talk briefly about a similar topic.
Thank you. Can you hear me okay? And see my slides as well?
Yep.
Thank you. So just to follow on what Will has said so far, and you might have heard him talking in particular about the overlap of some of the conditions that he diagnosed also with other rare neuromuscular diseases, and just wanted to highlight how this multidisciplinary approach is really key also for other similar rare neuromuscular diseases, and what is currently happening in terms of diagnosis and research for this condition, in particular for these 4 categories of rare and very rare ultra-rare neuromuscular condition which are the congenital muscular dystrophies and myopathies, congenital myasthenic syndrome, limb-girdle muscular dystrophies, and skeletal channelopathies. And the reason why a highly specialized MDT is very important also for this condition, because really there are many bottlenecks in the diagnostic process of these patients. And this is one of the reasons why this one approach, a single approach, is really not working or actually might affect the diagnostic abilities for this condition. These are very phenotypically heterogeneous. There is major interfamilial variability. And many genes involved in each supergroup of this condition, and again, multiple inheritance patterns, complex pathomechanisms, and often loss of function variants are actually not even pathogenic in this condition. Many private and missense variants that are often extremely complex to interpret and can be completely also missed and unfiltered. And also many of the genes involved in this condition are probably the biggest genes that we have in our genome, including titin or nebulin, with often complex replicated regions. And this is also why years ago, the highly specialized services has been set up also for this condition. So there are 4 services in 3 different centers in the UK. And each of these services offer an MDT approach for this condition with clinical, pathological, where applicable, molecular testing, and also functional interpretation for the variants. Now, interestingly, none of these services are offering testing through whole genome yet, because more specialized testing with custom-made small panels or custom whole exomes is currently still the best approach that best fits the purpose of these services. And indeed, our service, which is the one for congenital myopathies and muscular dystrophies in London, has been able with this triple approach between clinical, pathological, and genetic service to be able to actually offer quite a good diagnostic service for patients with this condition. And indeed, if you only look at patients who only receive genetic testing versus those who also have clinical review, with the MDT approach, the diagnostic ability really is increased. And this complex approach is really supported by a very strong body of evidence from international international centers as well. So why this is the case? So why do we think it's important? So clearly we feel that once we confirm a diagnosis, a highly specialized service can also help to timely access best treatment and best care for these patients. I also think that a more directed and filtered approach can be better to avoid misdiagnosis. Also, some larger panels can have predictive nature and test genes that you shouldn't really be testing in young children and avoid incidental findings. But most importantly, a specialist approach can help to improve understanding of interpretation of unclear variants with reverse phenotyping and offering functional analysis of variants through the highly specialized services, which is available and can be done for many of the genes that are currently being studied, which unfortunately is not yet available through the genomic services or other clinical services in the UK. And also plan better for those patients who are still undiagnosed, consider genes that are not classified as green in the panel genes, as we said, on analyzing genes that are classed as amber, and maybe plan other analyses that could be done either through highly specialized services or through NHS England approaches. And perhaps also consider other investigation could be done perhaps on research levels. So overall, I think these are the main reasons which really strongly supports what has Will already mentioned, why we feel that this highly specialized approach is key for these ultra rare conditions, and really all together with the different specialists involved in the services. And if any of you is involved in clinical activity or diagnostic with that are involved for the genes that are causing any of these conditions, please feel free to contact us, and we're more than happy to support or help or advise about further diagnostic of activity for these conditions. Thank you.
Okay, thank you, Anna. Thank you, Will. Really great talk. Really interesting stuff, and I mean, so impactful. I mean, you're basically doubling the diagnosis rate. That's amazing. Let me just check the question. So Sophie had a question actually that I was thinking about myself. She said, very glad to see great value added by MDT meetings. Did you estimate the rough time your team spent on each single MDT case?
We didn't time it, but it was highly variable, to be honest with you. I think it would have been from roughly a half an hour to an hour to maybe 2 days for some patients. So it really depended on what we found and also how strong the phenotype was. But I think manpower or woman power and having the time to do this is one of the most difficult bottlenecks. And having things like highly specialized services where you have a few more staff dedicated in this area makes it a bit easier. I think if you're in a mainstream clinic, say a neurology clinic or something like that, it's really, really hard to do this.
That time, yeah. Thanks. So Catherine asks, she says, great presentation, thank you. Are you reporting these new diagnoses through the diagnostic discovery pathway provided by clinical research interface at GEL?
Yeah, so I think some of them have already been reported by our clinical lab staff, but we are still in an early enough stage with some of the more research-based diagnoses, so they haven't all gotten through yet, but hopefully soon when we get enough functional data back to believe that they're true, they will be, yeah.
Thank you. So I think we got a couple more and we have a little bit of time, so we can work through. Hannah, if functional confirmation is needed to confirm genetic diagnosis of, say, a heart VUS, is there an argument that it should be funded via commission services rather than rely on research funding?
Yeah, I certainly think that is that there's a strong argument for that. So what we can't say, we can't go to the NHS and say that in-depth functional analysis for new genes and everything is a realistic expectation, but we can say that some simple things should be offered. So I think some genomic laboratory hubs offer basic RNA work for a start, and then some highly specialized services have some funding to do more basic analysis. For example, we do respiratory chain analysis, etc. in our group. In the muscle services in Great Ormond Street, they do a lot of stuff with different interpretations with their pathologists, different work on collagen and stuff like that, I think. So there's, I think for more straightforward stuff, we should have it available. And I hope that in the future with improvements in genome saturation assays and stuff like that, that it will become more straightforward. But there's an element for both, I think. In our centre, we have been very lucky that the National Brain Appeal have funded a translational scientist to do both genetic and biochemical confirmation. But we know not every centre is as lucky to have an extra member of staff to do that. I don't know, Anna, if you wanted to add anything in.
I wanted just to add that, yes, I think it's also the question that perhaps there is not enough knowledge, but nobody's fault, but about what is available in the different centers. So perhaps just raising awareness of what are the services already provided by specialist services, not just ours, but from other, that could help to aid diagnosis for patients and validate variants. So not just our service, but there are functional tests that are being offered through the NHS.
Let's see what else is on here. Andreas, do you work on ALS MND in gel? I don't know if that's a question for you or us. I think it's you.
We don't normally. We normally kind of stay away from those genes, but we do sometimes have patients with motor neuronopathies that can resemble them, but they're not true MND ALS genes, I don't think.
Najib says, I have a rather basic question. At which stage do you decide that this patient needed to go through mitochondrial testing?
So that's not a basic question at all. It's quite hard to decide who has mitochondrial disease and who should have it investigated. It depends wholly on their phenotype and family history in the first instance. So if you're not sure, even if you're a clinician and you wanted to decide whether or not you should do the testing, we're always happy to take an email and review the case and tell you if there's a likelihood. Because as Anna touched on, if you do this genetic testing, it's easy enough to come up with the VUS. And if you didn't really think they had primary mitochondrial disease in the first place, that's a bit of a nightmare. So it's based on the phenotype. And if you're not sure, we're always happy to take questions even before the testing has started.
And I think if I just may add on this, that because of the whole genome at the moment, there is not the possibility of reanalysis. I think before we do a test that might block the possibility of the patient to access new testing, that would be, the services are here to advise remotely. So to avoid an unnecessary test that might take maybe 6 months or longer.
Yeah. Okay, we'll take one or two very quick ones again. I think you're left. So let's see. Hannah has a follow-up question for you, Anna. She said, should we not test the whole genome sequences to enable future discovery research, but keep the analysis within the highly specialist services?
Should we not? Sorry, I'm just having a look at the
thank
question.
you for an overview of the services.
Yeah, should we not test the whole? Okay, well, yes, the problem, well, not the problem. Clearly, the whole genome analysis is great in it has a massive value, and I think that will be the future. But at the moment, because of the limitation of non being possible to resequence panels when one panel is negative, and the filtering approach that is currently set up, and I think currently we still prefer to go through custom panels, but I think that will be the future, to do whole genome for everybody once the process is set up for some rare conditions like these ones.
Okay, thank you. And then we have like 2 minutes left, so I'm going to take Alistair's question really quickly. Taking the 19 example, could it have been prioritized by Splice AI or by ClinVar annotation if analysis is done at a higher throughput?
Yeah, so Splice AI, and there's a couple of new tools, Squirrels and SpliceVault, are helpful, and I did use them. The issue is that for a lot of these, for what Anna is talking about, is that the filtering that we use via 100,000 genomes, it knocks out an awful lot of stuff that you want to know about. And so the single hit didn't even come to our attention because it was using a mode of inheritance filtering following a recessive path, and it didn't include the intronic one. So yes, in a broader sense, using these technologies for prioritization is really good, but you have to be really sure that the filtering pathway is suitable for the patient in the first place.
I think these filtering are good for the low-hanging fruit, so the easy, obvious mutation, common variants, but we're talking about condition where very often we have private changes, variants that are often also missense that might be easily unfiltered.
Yeah, makes sense. Okay, I think we're out of time, but I'll just say thank you. Thank you, Will. Thank you, Anna, for a really
Thank you.
very beautiful, clear talk and really impactful. And Wei Wei too, thank you. Yeah, it was great.
Thank
Okay,
you.
thank you very much. We'll see you soon.
Thank you. Thanks very
Bye,
much.
everyone.